Welcome everybody to the SynBioBeta Town Hall on the COVID-19 current epidemic. Let's wait a few minutes for everybody to join the town hall. I'm joined by some wonderful guests this morning: Megan Palmer from Stanford University, Jake Glanville from Distributed Bio, and Trevor from Mammoth Biosciences or Mammoth Diagnostics. We're going to wait for a few minutes for everybody to join, and it's going to be an interactive webinar. So we're going to have a few opening comments from our panelists, and then we're going to open it up to questions that we have on the Zoom platform. So you can click that Q&A button and you can ask questions, and you can also ask questions for the panelists, and you can also chat with us. And we'd like to test the chat feature. If you want to type in the chat box where you're dialing in from, then it will be great to see the diversity of people that we have on the webinar today. So as I said, for those who just joined, my name is John Cumbers, and I'm the founder and the CEO of SynBioBeta. And SynBioBeta is an innovation network for the synthetic biology industry. And the synthetic biology industry is a group of passionate individuals and companies and entrepreneurs and investors who are trying to make biology easier to engineer. And what better way than engineering biology than for rapid response to the current COVID-19 crisis that we're seeing. So we've got we've got a whole group of people from all around the world: Saudi Arabia, Portland, Guatemala, San Francisco, San Diego, Woodside, Esther Dyson in New York. Good morning, Esther, or good afternoon. We've got Warwick in the UK, Seattle, Vienna, Austria, UC Davis. More from New York, more from Danville. That's not too close from where I am. We're recording live from Lafayette, California. So wonderful, excellent. Stephen Hubbard from Zymergen, great. Carl Handelsman from Roche Venture Fund. Terry, hi Terry from Mintz Levin, the law firm. Louis Ceze and Karen Strauss from Microsoft in Seattle. So, wow, what a fantastic group we've got. So we're going to dive right in. Megan, I thought it would be good to start with you. Maybe you could just introduce yourself and what you do at Stanford and what your affiliation is, and then talk a little bit about the work that you've been doing before this latest virus hit, and then talk a little bit about what you're seeing at the federal level. Particularly, there was a funding bill yesterday. What's happening with that funding, and what's the current response looking like?
Sure. Thanks, John, and thanks for convening this discussion today. I think it's great to see so many people involved in stepping up and asking what they can do in this moment. So my name is Megan Palmer. I'm currently a senior research scholar at the Center for International Security and Cooperation, CSAC, at Stanford University. It's part of the Freeman Spogli Institute for International Affairs. The group at Stanford works on how we contend with the responsible development of biotechnology, including issues of safety and security. And I help lead some of the efforts around biosecurity at Stanford. There's several different groups. Mostly my group works on prevention and less on response. And I'm definitely not an epidemiologist or public health expert, but I can speak to some of the challenges connecting policy, technology, business in this space, especially around global challenges, and can direct to other efforts of my colleagues at Stanford and elsewhere that are looking at particular aspects of this challenge. I was actually just in the US Congress earlier on this week talking about leadership in engineering biology that did touch upon some of these subjects. How do we develop the types of technologies and platforms that allow us to be more prepared, allow us to both prevent and respond to these types of challenges? And certainly coronavirus is one of the things on the agenda. Our briefing on Tuesday, our hearing rather on Tuesday actually got pushed up because of a separate hearing on coronavirus, and there's been a lot of activity this week. So just to give a little bit of a landscape of what's going on, there's a lot changing. So we've now seen, sickening with COVID-19 of over 100,000 people worldwide, killing at least 3,400 people. There's a lot of effort to get the latest statistics. More than 300 cases in the US, although there's a big testing issue that we'll, I'm sure, speak about. And we don't have any FDA approved vaccines or therapies yet, although I think the panelists will also speak to where we are right now. Just yesterday, there was a funding bill passed in the US, HR 6074, and that allots $8.3 billion for response as a start. Just to give an idea of where that funding is going to go, there's a nice piece in NPR this morning or yesterday that outlines some of the funding allotment here in the US. So we saw 3.1 billion go to the Office of the Secretary of HHS, which gives some flexibility on how that funding is distributed, including 100 million to community health centers, 950 million to state and local health departments distributed by a CDC, with half of that to go out in the next 30 days. Good for mitigation, staffing, other types of analytics and surveillance. 300 million for buying vaccines and treatments, and some provisions around fair pricing. 1.25 billion for international activities. It's really critical in how we engage with the many, many countries that are also facing the same type of response. And then there's some additional details to look at when it comes to reimbursements that aren't covered here. But it's a really complex challenge. I want to highlight this different funding allotment because it speaks to the amount of money needed, the lack of funds in many cases for preparedness and response, and then the number of different places that this funding needs to go to in order to be distributed. I'm happy to speak a little bit later to some of the technologies that we might imagine for mitigating pandemic response and companies that are working in this space, and some particular issues around testing and authorization for testing. That's a key issue right now. But generally, I think we're seeing the emergence of a number of different coalitions in different technology and policy spaces that is making me hopeful to see some concerted efforts and coordinated efforts.
Great.
Is that enough,
Excellent.
John?
That's awesome. Thank you, Megan. And so the flow of this town hall, we got this overview from Megan just now, and now we're going to move on to Trevor Martin, who's going to talk about testing and diagnostics. And then we're going to go to Jake Glanville to talk about therapies and vaccines and small molecules. So that's the flow of it. We do welcome live Q&A. So if you have a question, you can type it into the Q&A box. I want to welcome some additional guests. I see Jay Metcalf from Neolife and Ian Hayden up at UW. We've got Hello Tomorrow dialing in from Paris and Jason Kelly from Ginkgo Bioworks. So hi to everybody. Thanks for joining us this morning. So I'm going to now pass over to Trevor Martin, who's at Mammoth Diagnostics. And Trevor, if you can give an overview of what the current situation is regarding testing for COVID-19 and what the issues are around testing. And specifically then, if you could talk about the white paper that you published a couple of weeks ago on how you're using this new CRISPR technology that came out of your lab for rapid diagnostics.
Sure. So I think on the diagnostic side, and that was a great intro, there's kind of two main areas that I think are interesting to chat about. So one is the availability of testing, just period. And then the second part is what type of testing is available and kind of what formats those are available in. And of course, kind of where it's available. So on the first one, I think one thing that's really interesting is that I don't think anyone would argue that the US got caught a bit flat-footed in terms of just the availability of testing generally. And you can go back and make a lot of stories about why that was the case. Obviously, there was some issues with some of the initial testing kits from the CDC, for example. And I think it would be good at some point to do a retrospective on how that could have been prevented. But then more broadly, even if those kits were all great, I think that there's just kind of a lack of available tests for many of these labs. And going towards one of the points Megan made about the regulatory framework, I think that was a big part of the impetus for actually some recent guidance the FDA give around what type of testing is possible. And in particular, they've really kind of opened the door to individual labs to validate their own tests that they have internally. And that doesn't mean they can go ship them out to other labs even, but if they have some sort of in-house test that they have validated for coronavirus, now the FDA is saying that you can use that to start testing. And I think the hope is that that means that now tens of thousands of labs around the United States will move forward with their own individual tests, and that'll kind of, in a distributed, very interesting way, very rapidly create tests that we can use in different communities for coronavirus. So I think that's been a place where it's very interesting that the government has stepped in and I think had an impact in terms of making tests available. We'll see in the next few weeks how many labs are actually doing this and how much of an increase in testing we're actually going to get. But even with that, I think there is a need more generally for just more tests. And this also leads back into just our testing strategy, right? So as the disease has evolved and kind of what we've seen has evolved, We've gone from a strategy of testing maybe only people that have been in certain countries in the last 2 weeks, like say China, to maybe anyone that's been in certain countries for the last 2 weeks and has been in contact with, or has been in contact with someone who's been in those countries, to the now where we're seeing community transmission of unknown origin in multiple locations in the United States. And I think that's a signal that we need to change our testing strategy. And I think that's where even more tests will become, maybe millions of tests would be necessary rather than say tens of thousands of tests.
And Trevor, if you can just step back a second for people who aren't familiar with the healthcare system or the diagnostics market, why is testing such a big deal? Why can't you just go off and order a test? Why can't you go to CVS? Why can't you just do a test at home? What makes testing so regulated?
Yeah, so we want to have a quality test, right? Because if we have random testing, that can actually be worse than no testing at all, because then you maybe drive fear, you drive people going into quarantine that don't need to, you start getting a poor understanding of where the disease actually is. So you kind of have these two things that you need to balance very carefully. One is that you want to make sure you have extremely high quality results, and the other one is you want to make sure testing is very available and accessible. And I think that right now, this kind of guidance by the FDA to CLIA labs is very interesting in that regard, because CLIA labs are obviously very regulated, have high quality standards, and will allow them to develop these tests internally. I think on the second part, though, that kind of leads into what you were saying as well in terms of the availability. I think what this pandemic has exposed is this huge gap in the molecular diagnostics industry generally, where we don't have rapid molecular, highly sensitive, highly specific nucleic acid detection just available everywhere. Like, we can't very rapidly set this up in airports today. We can't rapidly set it up in the field. That's not to say there aren't diagnostic technologies that can't go there. It's just that typically they suffer from sensitivity and specificity issues relative to, say, a test you'd use in a CLIA lab. I think that shows that for this pandemic now, we can do what we can to make testing more widely available, like quickly spin up really awesome synthetic biology technologies. But I think long-term, it's a wake-up call in many ways that we really need to invest in this ability to do rapid, decentralized molecular testing at scale, so that the next time when COVID 2021, we're not caught as flat-footed.
And we'll come back to vaccines, or we'll come on to vaccines in a second when we talk to Jake. But why can't we just start to test for all kinds of viruses, all kinds of flu? So instead of just sitting there moping around at home when you get a seasonal cold, you actually we're on it, we're testing for it, and we have a response network that's automatically in place. From as part of our regular healthcare system.
I think with synthetic biology tools, that future is possible. I mean, that's what we're all about at Mammoth. And I think that's what's exciting on the one hand, is that I see the future where we can get there. And I think that it requires, well, first of all, the whole synthetic biology community, not just one company or one individual or something like that. But also, I think this is a huge opportunity for the government to step in and say, we take biological surveillance seriously. This is just as important as preparedness for earthquakes or tornadoes or anything like that. And I think, it's really a good opportunity for us to assess, like, do we take these types of pandemics seriously, and are we going to prepare seriously?
Great. I'll let's come back to your white paper in a second. I want to bring you in, Jake, because Trevor's talking about a wonderful market for diagnostics for flu. He could create that. You'll then put him out of business with a universal vaccine. So can you give a little bit background to who you are, what Distributed Bio does, and talk a little bit about the universal vaccine technology that you're working on?
Sure. So thanks for having me on. I'm Jacob Glanville, the CEO, founder, and president of Distributed Bio. We're a computational immune engineering group. We work with about 50 different companies to develop antibody therapeutics, CAR-T, VHH, and other platforms. And internally,
what's VHH, sorry?
Single domain. They're tiny little antibodies that are produced by camels, llamas, and alpacas, and they're useful for diagnostics as well as certain types of therapeutic applications.
And what's HH? What does it actually stand for?
The VHH is just the nomenclature for a nanobody, which is a small antibody that's produced by the camelids.
Great.
Cool.
Excellent.
So we produce these kinds of therapeutics, and we use high throughput data acquisition to help guide our discovery of antibodies and look into the immune systems of individuals using genomic sequencing instruments to look at hundreds of millions of antibodies rather than the 25,000 odd genes, which gives us some insights also on broad spectrum vaccine design. So we've worked with the Gates Foundation on creating a broad spectrum vaccine for influenza. We are applying that same technology towards some other rapidly mutating pathogens, although in the case of the COVID-19, we're not working on a vaccine there. We're working on a monoclonal. So what I'm going to do is describe a little bit of the the state of the art on where we are with respect to a therapeutic towards the COVID-19 outbreak. So at this point, we do not have an existing therapy, but the good news is that biotechnology has never been better than it is now at being able to develop therapies quickly. We're better with HIV than we were historically. We're better with Ebola than we were with HIV, and we're better now than we were with Ebola. There's sort of 3 tiers of types of medicines that are currently being worked on. The first tier and the fastest is the repurposing of existing drugs towards COVID-19. So there's a set of small molecules. These are the kinds of pills you take that could be like aspirin or Viagra or Lipitor. And they tend to be antivirals or antipathogen drugs that were tested and determined to slow the growth of the COVID-19 causing virus. So Gilead is working on a drug that's called remdesivir. It was attempted to be developed for Ebola. It didn't work that well there, but they have reasons to suspect it may work better here. And that's being tested in humans already in China. And there are also tests being set up in the United States. And we should get those results in April. There's also an antiviral cocktail for HIV that, because of the way it works, the way those cocktail components work, there's reason to believe it could be effective in a very different type of virus. And there's some proof, at least in a petri dish, that that's happening. So all of the cases showed if you take that drug, you mix it with the virus in a cell that it could normally affect in a petri dish, it's slowing the infection. And then the third category is a chloroquine. So it's a 70-year-old medicine for treatment of malaria that surprisingly had some effects. Again, there's some reasons to be suspicious about whether that's going to play out in humans, but all 3 of these things are being tested in humans. And we should have some answers there in April. That's our best bet for having a working therapeutic, to have an active antiviral that's being repurposed, because that stuff has already been validated previously. It's already been tested in humans, and you can deploy a small molecule pretty quickly. The next category, which has received a lot of media attention, is the vaccine. There are multiple groups working here, and one of the particularly exciting updates was a company called Moderna used a RNA-driven vaccine. They're able to go through this GMP manufacture process astonishingly quickly in about 41 days. The challenge is that the vaccines have not yet been tested in humans, and testing of vaccines takes longer than testing of a drug you could give to a patient. Because a vaccine needs time to build up antibodies in the patient, which means you need to give them shots and then wait a few weeks, probably give them some boosts. So you're talking about 6 to 9 weeks out where the person would actually build up enough immunity after they receive the vaccine for it to be protective. And that means you can't give it to people who are already sick, because by the time they'd have the protection, they'd either be cured or they'd be deceased. So you have to give it to healthy individuals. You have to wait an extended period of time, at least when you're looking for efficacy, to see whether there's a drop in those that get sick. So that work's exciting. There's a quote that it'll be 18 months before it could be distributed more widely. And that's assuming that we're not going through all 3 of the standard phase trials that are typical for drug development. The idea is to get to a large enough phase trial, you can justify compassionate use and distribute to a large number of individuals.
Wow. Okay, great.
The third category are monoclonals. And that's the work that I'm doing, and there are other groups working on this. And so the idea is that instead of receiving a vaccine and waiting for your body to produce antibodies, you skip the middleman and you just infuse the antibodies directly into the subject. There are some advantages here. One is that you can give an antibody like the small molecule to someone who is sick and treat them immediately, because you don't have to wait for the immune system to generate those antibodies. You can also give them to medical professionals. An antibody has a much longer half-life in the body compared to a small molecule drug. With a small molecule, you may have to take it a couple times a day. Whereas with an antibody, you could enjoy maybe 8 weeks of protection. So that could be something you give to hospital staff and military personnel, people that are working in hot zones, as well as those that are sick. The downside is that we require engineering to get there. So some groups are starting from scratch. They're taking blood from survivors or working from other technologies to produce antibodies. But then once you have a bunch of antibodies, you have to go test a bunch of them to try to see which ones neutralize the virus in a petri dish. So what we are doing at Distributed Bio is I'm trying to skip some of that time by going back to a panel of anti-SARS antibodies from 2002 that people spent years of research after the outbreak determining that these were heavily neutralizing, they could protect in vitro and in vivo, and we're repurposing them. So we're evolving them to meet the slightly evolved surface that they recognize on the new COVID-19 causing virus. So if that works, that's the fastest method to get a known neutralizer to readapt to the new surfaces, but I will caution that we are still in the process of determining whether that can be correct. One of the final things I'll say, which is a challenge about COVID-19, is that for vaccines, there's a risk of something called antibody-dependent enhancement. And this is a phenomenon whereby which it was established in cats for a coronavirus vaccine, that when cats received a coronavirus vaccine, they actually became more susceptible and they got more severe disease. This is also why a dengue vaccine was recently withdrawn. And what happens is when you get a good antibody response from a vaccine, your antibodies surround and smother the virus on all sides, blocking it from infecting you. And then your immune system can grab on the back of an antibody like a handle and they can take it and destroy it. A poor immune response will bind weakly, not completely covering the virus. And when then the immune system grabs those handles, the virus can fall off and infect the immune cell that grabs it. So it makes the disease worse because suddenly your immune cells are being infected as well as the traditional lung tissue. So that's a concern that is not really present in the same way for a monoclonal because everyone's getting the same antibody in the exact same way, but it is a concern with the vaccines. And we've had coronavirus vaccines that work great for chickens. We've had coronavirus vaccines that failed miserably for cats. And so the question is, are humans going to respond more like a chicken or a cat, to massively oversimplify the problem? But that's something we're looking at as we move forward here.
Great, excellent. So a reminder, this is a live town hall and we have a few questions that people have asked. I'm going to come on with the questions in a second. And what we're looking for at the end of this town hall are suggestions or ways that we as a synthetic biology community can help this response effort. So we want to try to hone the question specifically towards that or specifically to ideas that people might have. And I have a few people who might join us if you just put your hand up, if you have something that you'd like to say live, then I can unmute your microphone and you can either come on with video or just come on with a voice chat and ask the panelists that we have here your question directly. We have a question for you, Trevor. How much can Mammoth scale its manufacturing of testing kits in the next couple of months? To millions, tens of millions, hundreds of millions? And this is from Isaac Larkin. Maybe then you can also just touch on the white paper that you mentioned.
Yeah, sure. I'll start with the white paper because that leads directly into the question. So very early in this kind of outbreak, we took time to actually develop a COVID-19 diagnostic. And I think why that was important to us is because a big part of the CRISPR-based diagnostics that we're building at Mammoth is how rapidly you can spin these up. And what we saw, and you can see this in the white paper, is that within 2 weeks, we were able to actually successfully get a test that we were confident could detect COVID-19, which is extremely fast, and that's really exciting. So the other part of the white paper, though, was that we wanted to be open about exactly how CRISPR-based diagnostics can be used in this manner to detect COVID-19. And in particular, going towards the second point I made before around the type of testing that's available. This particular white paper pairs the CRISPR technology with lateral flow strips. And that allows you to use it in a more decentralized format. So it's not as high throughput, let's say, as if you were using it in a traditional lab setting with robots that are processing everything. But it does mean, and we actually in the white paper, and I encourage everyone to check it out at www.mammoth.bio, and it's on our blog, and we can send out a link in the chat maybe as well. And if you look in there, we have actually a picture of everything that you need to actually run the COVID-19 diagnostic, and it's not much. There's no like heavy equipment or anything like that. It's just very simple equipment, many of the things that can be gotten from any lab supply store. And we're constantly simplifying the test as well. And it was important to us to open source this so that community labs and academic centers could see this and build on it. And we really want to kind of let 1,000 minds think about it as this epidemic continues to grow into different communities. We don't want to be the bottleneck on access to this type of technology. So that being said, towards your point, right, how fast can you scale? And because the test is so simple, you can actually excitingly scale very quickly, potentially. It really comes down to a lot of supply chain components in terms of, obviously we're seeing supply chain disruption now in certain areas of China. But I think in general, what's exciting is that from our perspective, really the components of this are unlikely to be disrupted in any kind of current supply chain problems. So that means you could, in theory, scale, if needed, to potentially millions of tests over months, which is definitely exciting, but that's the kind of thing where having institutions like the government step in and accelerate these things could be beneficial. Or, of course, at Mammoth, we're always interested in partnering with entities that are interested in this type of test. And of course, we're already in conversations, but that's something where I think there's an opportunity for the synthetic biology community to come together and partner and really scale these types of technologies like the one we're developing at Mammoth.
Great. And we have a question from Michael who wants to know, why can't we just use the tests that are in countries like South Korea at the moment and bring them here?
Yeah, so there's lots of different tests kind of all over the world, and I think that comes down to a couple of parts. So one is just the access to the tests. It's kind of sounds silly maybe in the 2020 that still, just where the tests are physically located can be a thing, but that's definitely
There's a lot of spare airline capacity now. They could just stick on a plane.
Exactly. But the second part is regulatory, and that's that if these tests are developed in other countries, they still need to be validated in the US, either at like a certain CLIA lab or by the FDA themselves.
And Tom Knight has a question. Tom, from one of the founders of Ginkgo. Good afternoon on the East Coast there, Tom. Tom wants to know what about environmental testing of samples? And I guess this is this thing can be transferred by by animals in the environment as well. So what's the situation around environmental testing?
Yeah. So in terms of environmental testing, I think there's not enough tests at the moment, maybe for individuals that even have potentially coronavirus. So I think, frankly, that's not being explored as much, but I think it's a good point, right? Like, especially as we see community transmission, it becomes all the more important to do more biosurveillance rather than specific testing only for individuals that are ultra high risk. So I think that's an area that, needs to be explored more.
Yeah, and Tom's also wondering what percentage of the tests that are done then result in sequencing of the virus, which would allow us to look at epidemiological spread.
Yeah, that's a good question. And I don't know the exact percentage. There might be something online where you could look it up. But I think that there has been a lot of exciting work there. And that's how we're seeing what things are community spread, where these diseases are actually originating. I'd have to look it up. But there's been some really great work out of the University of Washington where they have sequenced the strains of COVID-19 and actually have created the trees trying to show where the specific strains are related or not related. So there's definitely lots of active work going on there.
Great. And last one on the CRISPR technology from Ellen. CRISPR tech like Cas13-based Sherlock, that's another company on the East Coast, shows great promise. What's the IP barrier to other labs developing tests based on IP protected tech? Is it lifted in the case of COVID-19?
Yeah, so I think the idea here is that, and I can't speak for other companies, but at Mammoth, we're excited for people to build on the tech. And we can figure out the IP situation, all these things going forward. But definitely, we put the white paper out there so that people can come together, think about how this technology is best used. First and foremost, think about how can we get this technology to the people that need it. And second, we can think about everything else.
Great, excellent. I have a couple of questions for you, Jake, on the antibody development. These are from Isaac Larkin again. What's the estimated time to mass manufacturing of therapeutic anti-COVID-19 antibodies? What are the rate-limiting steps for developing antibodies, nanobody COVID-19 monoclonal inhibitors, screening, validating, or scaling recombinant products? And then also, what other protein inhibitors of COVID-19 are out there?
So thank you. This really speaks to why we're having this conversation about how synthetic biology could accelerate this process. Normally, the stages of delay are first in finding a drug that works, so the engineering and R&D. Next, on doing GMP and scale-up manufacture. Then it's the phase trials. So with respect to the first stage, we are going to know by the week of April 6th whether we successfully crossed the 2002 antibodies. We have 5 different antibodies, and we're working to cross them. They need to be high affinity picomolar binders that cross to the new SARS-CoV-2. It's about 74% identity in the receptor binding domain, so we think we have a pretty good chance on all 5 of those antibodies. But again, what we're doing is throwing 5 billion pieces of spaghetti at a wall and seeing what sticks. So our timelines are engineering will be complete by early April. The next period, which can normally, in an unrushed normal therapeutic development in a biologic, could take 18 months, which is GMP manufacture. But that's where there's been some pretty exciting advances in synthetic biology as well. So we're partnering with a company called SwiftScale, and we're also talking with a company called Angstrom, and both of these groups have these super accelerated methods for rapid batch GMP manufacture. One of them using a CHO development line. They were trying to create a personalized medicine solution to be able to make personalized batches GMP grade for treatment of people with anti-idiotypic targeting therapy for B-cell lymphoma. And they spent millions of dollars on building up that platform. And then the second group, so SwiftScale, who they've been great. So we started partnering with them, and they actually sent over additional scientists to accelerate my engineering side of the program. So I felt like the elves were coming over like to aid the humans against Mordor. They have an ultra rapid cell-free method, and then a couple of types of heavily engineered cell lines that enable this ultra rapid scale up. In that application, we're using single chain FV-FC, which is a form of antibody that we can express quickly in non-mammalian cell systems. So their thing also takes under a month to be able to develop. At that point, you have some regulatory hurdles that these groups have worked out to various degrees, but that material at that point is GMP grade and it's ready to move forward. Then into the phase trials, traditionally you do a phase 1, which is like 20 healthy people to make sure your drug doesn't hurt them. Then phase 2, which is a bigger group of like 200 or 600 people to make sure your drug actually has some efficacy, some statistically sound evidence that it works. And then a phase 3 trial is this massive trial to make sure it works on a broader population. You aren't detecting weird glitches in rare subjects. And then after that, it gets approved. Here, there are acceleration opportunities. One is that the cancer community has showed us we can combine a phase 1/2 in high-risk populations where instead of treating 20 people, you treat a larger number of people who are at risk for the disease in your phase 1. And so we'd be able to do that here in hospitals and offer, you can't do it with a vaccine, but you can do it with monoclonals, you can do it with small molecules. So you spend more money, but you end up getting information on efficacy in your phase 1/2. And then in phase 2, we've learned from the Ebola trials have shown that your goal is actually not to make an approved product. Your goal is to treat people during that period, which means you can spend more money in an earlier phase period, like 2 or 3, to be able to treat not just like 600 people, but you could treat thousands of people in a rolling protocol. And so those are ways you could potentially have this treating decent numbers of people by September.
Excellent. Ian Hayden, if you want to raise your hand, I can call on you to ask your question live. And we also have a question from Ari Lippman down in LA. Is there any information on how the government funding, the 8.3 billion will be deployed? A typical grant SBIR type mechanism will be too slow. Does anyone know of other funding sources, mechanisms that have popped up? And then Sandor Shoiket says, for the SARS outbreak, DARPA launched the Blue Angel project for rapid scale-up vaccine production, including novel approaches like plant-based production. Is DARPA reaching out now for a new production technique in anticipation of having vaccines to produce them soon? Megan, any ideas on how that new money is going to be deployed and how some of the agencies like DARPA are springing into gear here?
Yeah, I just put in the comments section the link to the actual bill. And so you can see the appropriations and the supplemental funding to the number of different organizations. And this speaks to the complexity of the coordination challenge, as well as the different facets that are needed here, from diagnostics all the way through development of new therapies and just public health response. So there's supplemental funding to FDA, to CDC, to NIH, to public health and social services, as well as to the Small Business Administration, Department of State, USAID. So the funding is allocated across several different groups. That's why I highlighted at the beginning the fact that we really need many arms of the response in order to be effective. And so you can look through that particular bill. There's other types of authorizations that will be required. And I'm happy to provide some additional info and link to others who are closely tracking this, so others can find out how they can be involved. To some of the other topics as well, in terms of an international response, it's worth looking at the Coalition on Epidemic Preparedness and Innovations, which have done a lot of work in looking at both financing as well as coordination, who are looking at some of the challenges around licensing and partnering in these areas. So it's another good source of information.
Excellent. So I want to go to Ian. So Ian, you should now be able to talk. Can you hear us?
Yes. Hi, John.
Hey, welcome. How are you? So you're in Ground Zero. You're in Seattle. Tell us. Can you give us a little bit of background of who you are and what you do? And then we've also had a question from Lisa Rich, who's returned to Washington State today and is wondering about the deployment of test kits there. She says 10 people have died. Do pharmacies have it, or what's the situation in terms of testing? So maybe you can give us a view of what it's like on the ground there.
To the extent I know it, I'll start with a brief bio. So my name is Ian Hayden. My day job, I work at the University of Washington. I'm a public information specialist for the Institute for Protein Design, which is run by David Baker. So we're working on a lot of computational protein design, including a lot of stuff around COVID-19. We're working on therapeutic binding proteins, diagnostics, vaccines, and crowdsourcing binder design as well. As for the latter question, no, it's not in pharmacies. I do know that the University of Washington developed a version of its own real-time PCR-based test, and that is now analyzing samples from the Seattle area. They've got a couple automated robots to do that, so they're able to scale up. I think it's a couple hundred per day now. They estimate they could do 1,000 per day or perhaps more soon. And that was enabled by having those robots up and ready to go and getting FDA approval to run those tests in-house. I believe all of those positive samples still have to be routed through the CDC, but it has significantly increased testing capacity here locally at UW. And I think especially those who are getting tested are healthcare workers at the UW medicine system who've been in contact with patients.
Excellent, great, thank you. And I think talk a little bit about the vaccine. You were talking about, I think Vice President Pence was holding up a model of something that had been developed by the Baker Lab this week.
Well, that was Trump himself.
Okay.
So we've got a long-term partnership with the NIH for our computational vaccine platform, where the basic idea is we've made some protein nanoparticles, and we can stick antigenic proteins to the outside of that. So we were able to very quickly whip up a protein nanoparticle displaying multiple copies of the coronavirus spike protein on the outside. That was manufactured and stuck into animals very quickly. So that work is ongoing at the NIH right now. And as part of Trump's visit to the NIH the other day, he was briefed of our status of that trial, along with the other stuff that is being operated by the NIH.
Excellent, thank you. Ian, any other comments or any questions that you have?
I would just like to put a quick plug for Foldit here. So Foldit is a free game one can do on a desktop computer. And we've launched a new coronavirus puzzle, which lets anyone, including non-scientists, try their hand at designing a protein from scratch that would stick to the coronavirus spike protein. We've got a couple hundred players working on that now, soon to be 1,000, I think. And the most promising of those player solutions we intend to manufacture and test at the Institute for Protein Design and feed them into our testing pipeline.
Great, excellent. Thank you, Ian. Thanks for joining us. I hope you're enjoying the town hall.
It's been fun.
We have a lot more questions though to get through. If you would like to ask a question yourself, then just raise your hand in Zoom, and then I'll call on you to ask the question directly. Natalie Ma wants to know if we're integrating and sharing data across organizations, is there anything besides bioRxiv for coordinating and centralizing information? I don't know whether anybody has any thoughts there. Jake, you're on mute.
All right, yeah, I do have some thoughts on this point. I think it is, on one hand, been remarkable how effectively we've been able to share sequences, structures, and new information, but bioRxiv has been a problem here. What we have seen repeatedly in this outbreak is that people have posted work that has not undergone peer review, and it reaches the point of pseudoscience, and it's defeated, I would say, one of the major successes of the scientific method is that we have smart people review other smart people's work and decide whether it's fit for print. All of us make mistakes, and that's why we should all review things from multiple angles, and there's been lots of conspiracy theories and unvalidated reports that have made it up, and they bear the sheen of real science, but they have not gone through the process that makes that data trustworthy. So it's actually a detriment to the quality of science in an era where that is more important than ever before. So I actually am frustrated by bioRxiv right now and how it's being misused during this period. We want to have rapid data sharing, and groups have gone through traditional, very rapid review and managed to get their papers out in real journals. So that's not that can't be done. I think things should continue to go through the appropriate channels to get validated data that has undergone peer review out in people's hands. So that's just my opinion here, but I think I've been having to field questions about the outbreak, and I'm having to sift through a bunch of these weird reports that end up in the media, and I trace back to an unvalidated bioRxiv article. So that has been frustrating in my mind. Actually, it's sort of like a weird bug in the current well-meaning but unfortunate consequences of the bioRxiv as a place to post things that look like science but are not science yet.
And there's an interesting opportunity here, like you mentioned, for journals to step up and accelerate things, and also similar to how news outlets have taken the COVID-19 articles and put them outside of the paywall. I think there's a huge opportunity for journals to do that as well, and that could be a part of that problem.
I just want to say as well that, you know, you're bringing up an important point as well about just data sharing on samples generally, right? So there's CDC standards and others around data sharing and patient samples, and WHO's within this as well. But this is a much larger problem around how we think about both samples of pathogens as well as human samples and our data being sent elsewhere for testing. That is something that we really need to pay more attention to.
Excellent. So I want to go over to Jeremy Zucker. Jeremy, if you can introduce yourself and then ask your question, please.
Sure. Thank you. My name is Jeremy Zucker. I'm a computational biologist at Pacific Northwest National Lab. currently working on a project to curate viral pathogenesis, immune response to viral pathogenesis, and perform causal reasoning over those models. And so my question is, I'm a little new to virology, and so all this kind of jumped onto my plate. And so I just want to know how well understood are the molecular mechanisms of coronavirus infection as compared to, say, influenza? Is this something that a distributed team of curators combing the SARS, MERS, and the new COVID-19 literature could help with in terms of consolidating that understanding into a consensus model and defining current gaps?
So that's a perfect question for Jake, but his laptop just died, so he'll be joining us back in a minute. So why don't we pause that? Any other questions, Jeremy? So if not, we'll just wait for Jake to come back on, and then we can take that.
Sounds good.
Okay, Stephen Hubbard, you have a question as well. If you can just introduce yourself as well.
Can you hear me?
Yeah, we can. Go for it.
Okay, great. So I am curious about the current throughput of the qPCR testing that exists. I read that NPR article that you just linked, and it sounded like they were excited to do 4,000 samples a day, and that seems like a lot. But my company is working on technology to massively increase the throughput of laboratory automation for the types of things that we're currently doing. Haven't really thought about it for this type of test, but interest is growing there, and I'm wondering how I should counsel the automation engineers and the executives at my company to recommend that they devote some of the resources towards being helpful.
And Stephen, your day job is at Zymergen here in the Bay Area.
Correct.
So Trevor, do you want to take that one?
Yeah, definitely. And I think you make a good point, right? So like 10,000 tests sounds like a lot, but then when you put it in the context of the US population, it's woefully inadequate, right? But I think that's more of like a per lab. And I forget who made the point earlier, but at University of Washington, for example, certain labs do have their own internal automation, which is great because it means you can go from like hundreds to thousands to maybe approaching tens of thousands of tests per lab. Right. And the idea is that if you have 10,000 labs across the country, you start to get to real scale through that. I think there is an opportunity here, and companies like LabCorp and Quest, I assume, also have like pretty large automation facilities that could tackle this on even larger scale than any individual academic clinic. But I think in general, the key things we think about in terms of automation when we're thinking about diagnostics is that you have to really remove all the bottlenecks, right? So it's not even necessarily just the amplification step. What about the sample prep? Or maybe you don't need sample prep. Because if there's a single part of that process that's not automated, even the interpretation, right? That means that now all that automation that's built up maybe isn't as effective as it could be because you're still bottlenecking at one specific step. So I think
certainly, and then the platform that we've been working on automates all of those types of steps for all types of biological manipulation.
Right.
So I can see it being beneficial.
Great. No, so that's awesome, right? So it sounds like you've already thought through that. So every single step is automated. And then the next part of it is the validation, right? So a huge part of diagnostics and CLIA labs is the quality controls. And that can be very different from, the first time you spin it up to reproducible, validated, documented. And I think that's just as important as the technology of automation is the regulatory quality controls, reproducibility, because there's plenty of things that you can automate, but not reproducibly. So I think both.
Who do you think that we could partner with who would be knowledgeable about that validation step and knowledgeable about how to get these tests actually run in BSL-2 laboratories and HIPAA laboratories?
Yeah, so we'd be happy to chat with you about, our thoughts on this in more detail after the call. But in general, I think anyone that has CLIA lab experience could help you think through these different kind of issues that would arise as you try and automate diagnostic testing.
Cool. Thanks very much. I'll take the rest offline.
Great. Thanks, Stephen. And if either of you want to email me and you don't know each other, I can connect you. Jake, while your battery was being recharged, Jeremy Zucker had a question for you. How well understood are the molecular mechanisms of coronavirus infection as compared to influenza? Is this something that a distributed team of curators combing the SARS, MERS, and the new COVID-19 literature could help with in terms of consolidating that understanding into a consensus model and defining current gaps?
Sure. So the good news is that we have spent a lot of time on 2 trial runs, really getting to understand SARS and MERS on the outbreaks from 2002 and 2014. In addition, the coronavirus, the chassis, is remotely related to a set of viruses that cause what looks like the common cold. So we have a considerable amount of familiarity with the virus. Also, we develop vaccines for this virus, again, remote relatives of the virus, of coronaviruses that infect animals. So in that sense, we're familiar with them. The thing that is different that we constantly have to check is, and this is the case for any rapidly mutating virus, it's what is it using to gain entry into the host. So in the case of SARS, there was this ACE2 receptor that it used to gain entry, and we can see the new coronavirus apparently also uses that receptor. But because of a significant amount of mutation on a couple different loops on the receptor binding domain, I look at that and I immediately ask, okay, just because it binds to the SARS receptor, which is ACE2, that doesn't mean it doesn't have a second receptor. That's not unusual for viruses, and it could help explain why there's this unusual shift in the degree of severity of the disease of very young children versus old individuals that we did not see in SARS or MERS. So I think the missing piece there, unfortunately, it's biological. That really needs to be a massive search of the receptor binding domain against a bunch of other potential mediated receptors. And so I don't know if that's going to be accessible through like sequence analysis. I think it really is going to happen through wet lab analysis, but that's a worthwhile exercise. So with HIV, as soon as we found different receptors, we gained engineering insights into how to better block the disease from progression.
Great, excellent. So I've got two more people I want to call on live. Next is going to be Vikesh and then Isaac Larkin. But before I do that, I just want to whiz through. We have 19 unanswered questions. So I'm going to try to group a bunch of these together, throw them out there, then call on Vikesh and we can answer them. Louis Metzger about global access of putative therapeutic biologic targeting COVID-19. This would often be restricted to regions of the world with cold chain access. That is, they keep the antibody cold until it gets to the patient. Thus, there would still be a need for small molecule drug development, he says. Maria Chavez, if you want to ask your question live, I think it's a really good one. So just raise your hand and I'll call on you. Otherwise, I'll come back and ask it for you. Sarah Calvala says, how are we making sure that competitive interests, whether between countries or for-profit companies, is not going to hinder progress? So we can come back to that in a second. And Esther Dyson has a very practical question. I'd love to know more about the test in a practical way. As I understand it, you see a doctor to get authorized / prescribed a test. How is it done? How long does it take to get an answer? And how could that change? And I think that's quite related to your question, Vikesh. So if you want to ask your question now.
Hi, thank you everyone for having this wonderful session. My initial question is in the context of a diagnostic, given that these viruses have very conserved sequences, so before I go into the details, my background is that I have 25 years of experience in molecular biology and I'm an established cancer biologist as well.
Great, thank you. And which institutional company are you with, Vikesh?
I used to be with UCSF Medical Center. I was a postdoctoral fellow there, and now I'm a cancer biologist in the private sector.
Great, excellent, welcome.
And so one of the things that I wanted to discuss is that if these sequences within the viruses, SARS, MERS, or COVID-19 are unique and different from the human, then why not just use these sequences to do a rapid PCR, but not a qPCR. It doesn't even have to be a qPCR. You can just multiplex it without having to do you can do 3 different sequences at 3 different sites within the genome, and boom, you have your answer. These are rapid, quick methods to get around the bottleneck, and you can do this at scale. Just make the sequences available, use your primer sequence, and really go to it. But there's a tethering question to this. Unless you have a therapeutic, Jacob, like you're attempting to develop, what is the true value of these diagnostics other than for quarantine purposes? If people are going to get sick and they're exposed, what are we going to get from a diagnostic? It's the same issue we had with SARS and MERS. And as a background, I lost my aunt to H1N1 about 3 years ago, and a diagnostic really didn't tell us much when they diagnosed her.
So sorry, Vikesh. Jake, any thoughts?
Yeah, so I'm going to let most of the question of the value of diagnostics to the other panelists. But the punchline of diagnostics, if we don't have surveillance, then we don't know where the outbreaks are. And because there's a long delay of incubation, then we are unable to break the outbreak. So it's critical for us to have diagnostics in order to know how to suppress further outbreak. With respect to the therapeutics, I'm sorry, I got distracted. What was the question?
It's to tether a diagnostic, so have a companion diagnostic to your therapeutic.
Sorry, Trevor. Okay, so there is a value in doing it, but I really think Trevor should answer the questions about PCR. So go ahead, Trevor.
Yeah, so a bunch of parts to your question. So I'll start with the part just on the technology itself. So I think what you described is, in general, like a great idea. So the CDC's made, I believe, the primary sequences available. So there are primary sequences out there. I think the key thing there comes down to the reliability of the result and the sensitivity and specificity, right? So especially on the specificity side, I think that one interesting thing, essentially the way the white paper CRISPR-based diagnostic method that we put out there and that other people have put out there works is pairing a very simple isothermal amplification with the CRISPR technology. And that gives you a double amplification essentially with a huge increase in the specificity of the reaction from the CRISPR protein as well. So I think that in general, I would agree with the kind of high level approach of like, yeah, what if you had really rapid isothermal techniques that could tackle this? And I think the rub there is you really need to make sure that they are highly sensitive and highly specific as well, because there's a lot of coronaviruses out there and you want to make sure you're actually detecting the right one. And then the final part to that is the reliability of it. So the reason why you wouldn't necessarily just want to have any kind of random lab running their own testing facility without any oversight is that what if they, for whatever reason, are not running it correctly or reproducibly or reliably, or maybe their reagents just happen to be off. Then you start to, going back to the point that was made before, potentially start to think you have outbreaks as a place you don't, so you misallocate resources. And I think when you're developing to go more towards the second part of your question, so I'm not an expert on vaccine or these types of development, but it seems to me it would be critical actually to have diagnostics so that you're not, for example, giving a vaccine to someone that already has the disease. So when you're developing it, you're not getting confounded data. So even beyond the quarantine part, it seems like a diagnostic is critical for the therapeutic development, even just in practical respects.
Yeah, we would only give the antibody or a small molecule to someone who was COVID-19 positive because there's no purpose. Otherwise, you're wasting medicine in a period where you need it. Yeah.
Right.
And Trevor, if you could just answer Esther's question on the practicality of testing right now. How is it done? How long does it take to get an answer? And how could that change?
Yeah, so right now, the typical PCR test has at least a 24-hour turnaround. So the PCR itself, maybe 4 or 5 hours. In the white paper, we actually benchmark the speed of our test versus other available things like the CDC test, for example. And I think right now, all the tests are kind of being validated against each other in general, and people are trying different techniques. But the hope would be that the CRISPR test is actually much faster than the kind of existing PCR-based methods, so that you could actually get a result more quickly without having to wait as long. So right now, maybe you get the result 24 hours later. Ideally, it's like a full day process. Ideally, you could bump that down to just like a few hours, for example.
Excellent. And how is your test different from the qPCR test in terms of some practical differences?
Yeah, so in terms of practical differences, so in the white paper, you can see exactly what's going on, but one is that the readout is a lateral flow strip, for example, so that you don't need to have necessarily the same equipment that you would need to run these PCR-based tests and interpret them. And then the second part is that because it has the CRISPR technology in the loop, it's just a much faster process.
Great, excellent. Isaac Larkin, over to you if you want to quickly introduce yourself and ask your question.
Yeah, hi everybody. I'm a PhD candidate at Northwestern. This is sort of a question for, I guess, Megan. What efforts are being made to sort of implement collaborative and sort of centralized information sharing around all this stuff? Like, who are all the labs that can develop, diagnostics? All the companies. Who are all the labs and companies developing therapeutics, vaccines, and what are what stages of development are those? Like, is there a centralized, like, I don't know, a wiki, or is anyone working on building a resource where people who want to learn more about this sort of thing can go?
Yeah, I think that's a good point and good question. I'm not a public health expert, but my advice would be to look to the local public health experts, right? So here in the US, for instance, I think this also gets to Maria Chavez's questions about how you, engage locally, and you see county level public health are good sources of information, and you see coordination between different local, state, national constituencies. And so really, the best thing is to engage at the local level, and then to connect in so that you can connect with with the national level coordination capacities. And it speaks to, as much technology as we may have, we need to invest in building up the coordination and communications infrastructure, because that's really what we need in order to rapidly respond. We need to invest early, and then when something happens, which it inevitably will, right? This is not the last time we will face this type of outbreak. We need to get better at preparedness planning so that when something happens, we can have that type of information sharing. I think it's really important also with this whole community. And something you can do immediately is just make sure to redirect to legitimate sources of information, right? Again, county public health, CDC, WHO, and others, and not rely on these secondary sources of information that can often be flawed and have a lot of misinformation.
Great advice, Megan. So Maria, if I can just pass over to you to ask your question live. Can you hear me?
Yeah, I can. And I think Megan addressed this, but one of the things I was wondering about was just this idea of how do we engage our local communities more, because we're talking a lot about federal response, but I think anybody, no matter where you are in the country, wants to know what local policies should we be looking at engaging our communities in for preparedness, and what questions we make sure that our communities are engaged and prepared.
Great. And Maria, you're at BioCurious in Sunnyvale, is that right?
That's right.
Excellent. I think we have another few community labs on the town hall as well. So any thoughts from any of you in terms of engagement in your local areas?
Yeah, I mean, I think this is
go ahead.
No, Megan, you're better qualified.
No, I was just going to say on this direct topic, right? Santa Clara County Public Health has an information page that gives a lot of really good guidance about what you should be doing, especially with different types of vulnerable populations. And I think this is a moment with any type of response where engaging locally and figuring out how you prepare and take reasonable measures that are in accordance with what we know about risks is really important. So we don't all individually do poorly informed things, but collectively are responding and preparing. And so I would again just redirect to the legitimate sources of information that exist in your communities. It's great to have discussions comparing what's happening in different places, and some of the efforts of the community labs just have a collective learning about what's occurring, and are really great efforts.
One thing I would add here is I would encourage, this is me being politicking a bit, but I would encourage all communities to contact retirement homes and nursing homes and other groups that have high-risk individuals, and encourage them to set policies to limit visitors until surveillance is established. I'm actually surprised that this has not already been instituted at a national level. Once surveillance has been established and we know where the hot zones are, that's more effective. But in the meantime, call grandma, don't visit her, because that whole community is very susceptible towards large-scale infection.
And in general, I think this is a huge opportunity for education, because everyone knows what to do in an earthquake, what to do in a tornado, but no one knows what to do in a pandemic. And I think, like you said, this isn't going to be the last time that a disease like this comes along. And it's a huge opportunity for community labs and kind of sources of authority for science to really give people a science-based approach to how do you deal with something that they've really maybe never thought about before.
So Megan, I do have a question for you. Someone at my company received notice that there was a teacher at her school that was COVID-19 positive like a week prior. They had obviously had a series of questions, and they were a little bit surprised there had been no follow-up to any of the other teachers or students that they've been in contact with, because it's a series of schools in Santa Clara and multiple other areas in the Bay Area. I spent an hour and a half trying to call the CDC, and they never picked up, and she did as well. And I received messages. So I think, what do we do if these resources that should provide us guidance are inaccessible, maybe because too many people are contacting them?
Yeah, I can't speak to that particular case and the particular challenges of getting connected. I don't have first-hand experience, and I'm not directly connected in, but at least we see now there's a lot of relying upon also local institutions, right? There's been a lot of communication at Stanford with the medical school, others, right? Who can you rely upon? A lot of these things end up being hyper local, and a lot of this community, it's great that you were able to speak with many of these folks who may not know as much about the disease progression, what's reasonable, and so we all are really resources within our communities to try and give the best information. And if you can't, escalate to a higher level, try and find, the most valued, legitimate place within your local community. And that's what we really need to do in these moments is rely upon others in our community who have better information than we have as individuals.
Are we currently performing contact tracing in the United States? And if so, who's responsible for that?
Again, I'll have to redirect to this area because I'm mostly on prevention and lesson response. But we can follow up after this with perhaps a set of information about, at least in the U.S. and elsewhere, who you can seek for information. A lot of what I've heard recently is around, how do we redirect efforts now that we're seeing community transmission? It means that, instead of trying to quarantine, we're trying to put resources towards just making sure that those who are sick are getting the care that they need. And so this is changing very quickly. And I suspect it's going to change even more as we start to see tests rolled out and get a better idea of the real scale of the problem.
Absolutely agree. I think that getting diagnostics out there for having effective surveillance is just an essential first step. I think there have been some missteps getting here, but I feel like that's why I was very excited to hear Trevor's work. And just in general, finding some way to democratize that without compromising on quality, I think is an essential first step to be able to get a handle on this problem.
And I think just one last thing, just looking forward, again, this is not the last time we're going to have to deal with this. And so there's a lot we need to do in this moment, but we also need to make sure that we're advocating for preparedness at all levels. And we were caught flat-footed in many ways, and we didn't have to be.
Yeah, agreed.
Right? We didn't have to be.
Is there something you could suggest that we all do, communicating with representatives, or what pressures can we take to help us be more prepared next time?
Yeah, I think there's a lot of good guidance, and I'm happy to follow up with some of that afterwards on some of the efforts of coalition building around this. These are being activated now, but we need more.
Thank you.
Great. I want to be respectful to our panelists' time. We had 20 questions that we didn't get to, so we may do this again next week. I appreciate all of the panelists, Megan Palmer at Stanford and Trevor Martin at Mammoth Diagnostics and Jake Glanville at Distributed Bio. We had 150 people joining us live today, so this has been very valuable. And I'm sorry that we didn't get to all the questions. We might think of a different format so that we can allow other people to answer each other's questions in the format so that we can have a bit more interactive on this. But thank you all for joining. This has been really fantastic, and thanks to all of the work that you're all doing, and we will see you next time. So thanks again. Have a wonderful weekend, and stay safe and stay healthy. Thank you.
Thank you.
Thanks.